Welcome everyone. Thanks for coming. Today is week 7. It feels strange that we're already, in the final weeks of the class. It feels like we've just started, but we really are. In the not even the last quarter of the class almost. And we'll be this week and next week we'll be finishing up our second phase of the class. So we'll be this week, of course, sulfur and trace elements, and next week we'll be looking at poisons. And then in our last 2 weeks, we'll be concluding the course with a more like longitudinal view, looking at the fate of the elements as well as potential for artificial life, artificial in the sense of not like the life that we see here on Earth. But at this stage, we've finished phase 1 where we talked about the formation of the universe, we talked about the formation of elements, we even talked about how the elements came to be on Earth, as well as some of their uses, and as well as life in general, how we should think about it. And you guys had a great conversation about that. I was very pleasantly surprised to hear some of the ideas that you guys shared about the definition of life and so forth. Then we looked into some of the core elements for life. So far, we've discussed in depth oxygen, carbon, hydrogen is more of a filler element, a little bit less specifically focused on it, but also nitrogen and phosphorus. These are the core component of SHNOPS, the acronym everyone points to when they say these are the elements essential to life. But they kind of leave out a lot of the nuance, a lot of the really cool things about biology, about life in general. And I think that it's best to start at the very tail end of SHNOPS with sulfur. Sulfur almost is kind of like an oddball in this group, where we talked at length about carbon and nitrogen and oxygen, about amino acids, about the structures that they form. But I feel like in our general education that a biology major, say, might get at UCLA, sulfur is probably the least talked about. But it has some of the most interesting chemistry to do it. So with this presentation today, I hope to highlight some of those key points, and then from there we'll transition to some more trace elements. Intentionally, actually, I've only focused on a few trace elements in this lecture today, because you all have been putting together, or will start to put together, presentations focusing on other trace elements that are important to life. And I think that the justice you guys will do in that 10 to 15 minute presentation will outweigh me like rapid fire going through 15 different elements. So I'm going to only focus on a few trace elements with the knowledge that you all will do a wonderful job on your presentations. That said, let's get into sulfur. So the anatomy of sulfur is very interesting. It's the 5th most abundant element in the universe, 10th most on Earth. And as a result, it's found in a wide variety of materials, as well as sometimes by itself. We talked last week or 2 weeks ago about how some elements really don't jive well by themselves. Phosphorus is very volatile. Same thing with some elements that we haven't discussed, like potassium or even sodium are very volatile by themselves, and so they don't often exist in nature as themselves. They exist in the form of other compounds. Sulfur is kind of in between those two extremes, where it's able to be formed in pure form, and on Earth it's most often found in its pure form in neovolcanoes or hydrothermal vents for reasons that we will discuss later, both organic and inorganic reasons. But as also in the form of minerals. It does play well with other minerals, and one that you may be familiar with is pyrite, or fool's gold, which is a mineral composed of both iron and sulfur. And together they form this very lustrous but fragile, very brittle mineral that has been storied in history as, of course, fool's gold. But these are just some of the physical properties. The chemical properties of sulfur are very interesting as well, perhaps chiefly because of its location on the periodic table. So you will see sulfur number 16 right underneath oxygen. And just because it's right underneath oxygen doesn't mean much by itself, but some of the properties that oxygen enjoys and some of the unique characteristics can be passed down that column as well. Sulfur is twice as heavy as oxygen is. Its atomic weight is 32 instead of oxygen 16, and some of the same characteristics are followed through that. So when we look at sulfur, we also have to keep in mind its electronegativity and how that will factor into the reactions it's able to have. So because it's below oxygen, it's less electronegative, but it also has a very special property that we'll touch on in near the midpoint of our lecture, and that's its ability to have a larger spread of electrons around it. So in these two rows right here, in 1 and 2, I discussed earlier about the octet rule a few weeks ago, where these are elements that want to have 8 electrons around them. That's their most stable form, of course, other than this first row, which only wants 2. That, if for these rows, is what's considered most stable. It's the preferred form. And so these elements in this row are going to form associations, going to form molecules that are most stable when these elements have 8 electrons around them. But as we discussed earlier, everything in chemistry is alive, and that doesn't necessarily need to be the case when we go down one row. Because when we go down one row, we see that these elements are able to expand their octet. They're able to take in more than 8 electrons because they have access to another subshell of electrons called the D shell. Now, the reasons behind this really, I tried doing some research, and the best answer I can give you is because we assume the Schrodinger equation to be correct. That's why. That's why these can form an expanded octet and these can't. There are, of course, other reasons as well, but we'll discuss why it's important that sulfur can have an expanded octet and how life is able to take advantage of that fact to the fullest extent in a little bit. So, now that we've talked a little bit about sulfur in general, let's talk about how it first came to light as really important to our discussion today about how life in general. And I think the best place to start is in the field of pharmacology. Sulfur has actually been known to humans since antiquity. It was even known as brimstone in the old days, and so it has references in the Bible and other religious texts under that name as well. Hell was actually, for these individuals writing these religious texts, hell was thought to have smelt like sulfur because around the volcanoes where sulfur could be found, that smell of hydrogen sulfide was prevalent. But it was much, much later that we found a way to harness sulfur and actually bend it to our will. And I think that the story that I'm going to tell you, is just one of those stories in science where someone took a chance and changed the course of history. So right here, we're looking at a man whose name I cannot pronounce, but we'll try my best to, Gerhard Domgach. And he was, in the 1900s, a German bacteriologist. And he had a very storied career that we'll discuss in a second, but chiefly first, he was a family man, and he really prioritized his family, and that will really intersect with his story in a second. So he found himself interested in bacteriology at the very beginning of the field's foundation. The microscope had just recently been invented a few decades earlier. We had just now had the chance to look at bacteria, understand them, and kind of try to figure out what we can do about it. Because at this time in history, the simple infection, just a simple slash across the wrist, could be deadly, and often was very deadly. We didn't have the ability to protect ourselves from infection or to fight bacteria outside of our own immune systems, which, although are very good, and we enjoy some of the benefits of immunity still today, where we find ourselves in the pandemic, it's still not great, and it really has been bolstered by our modern innovations. And so, while working on a simple dye, actually, this chemical that we're going to be discussing, Pontosil, was first synthesized as a red dye, and that's all it was used for decades. Because we didn't have the technology, and we didn't have the wherewithal to see what its uses could be, or even really very carefully analyze its structure either. But in the 1920s, Domgach was responsible for experiments at his university, where he would infect different groups of mice with bacteria, and he would give doses like 10 times what's considered the lethal amount. Then he would give one group this dye, Pontosil, and he wouldn't treat the other group. And he saw through those experiments that the mice that were injected with the dye survived more often than the mice that weren't. So this was the first evidence for antibacterial activity of any drug that we had ever synthesized. And this was, I believe, in 1932 that this experiment was carried out. Now, science moves slowly. Science does move fast sometimes, but science does move slowly also. And it was many years of clinical trials that would prove the efficacy of this drug. But it turns out that Domcock didn't have that time, because in 1935, his daughter, while she was sewing, accidentally embedded the tip of her sewing needle into her hand, which began an infection that threatened her life. Even it progressed to the point where she had very high fevers, and the doctors at the time believed that the best course of action would be to amputate the arm in order to attempt to save his daughter. And there was really no guarantees even at that point. And Domcock couldn't take that. He believed that his dye that he was working on could be the saving grace for his daughter. And so he actually stole a vial of the dye from his workplace and used it on his daughter. And it wasn't like an immediate therapeutic benefit. She actually got worse before she got better, but she did get better. And this really gave this scientist the confidence that this is going to succeed. And as the clinical trials progressed, eventually in a few years, that therapeutic benefit was seen to be real. Now, of course, it's science is the story that I've been telling you over these past few years, or few weeks, I should say, aren't that simple. The story doesn't end there. You would naturally think that, oh, this man created an antibacterial drug, yay for the world, people can survive infections they would have otherwise died from. But it's a little bit more complicated. So the company that this man worked for is the same company, I believe it's called IGF, no relation to the antibody, that actually synthesized later. It was a German company that ended up during the war synthesizing Zyklon B. So it's a very, I feel like every week we end up talking about the Nazis for some reason, but that's just how it is. This period of history is really dominated by them, unfortunately. And so they, of course, they are bad actors. They forced Domcock to publish his results in a German-speaking publication, so that way it wouldn't be very widely publicized. It wouldn't be known to the rest of the world. So that way they could keep the patent and keep churning it out. And as a result, the drug flopped, and no one used it for many years. It wasn't until FDR Jr. The son of FDR, of course, the president of FDR, was infected and was saved. Actually, his life was saved by this drug, that it only really became popular, it became known widely. And that's when the rest of the Western medicine really picked up and began using it. Now, the patent, of course, still belonged to IGF, and no one really wanted to be at the whim, at the mercy of this company. So what they found instead, they did more research out of spite for this company, and they found that Prontosil was actually not the active agent in the drug. What actually happens is when it's ingested by the human body, is it gets broken down into a smaller chemical called sulfonamide. And that's really the structure that it has the antibacterial properties. And so they said, it's not Prontosil. That's these were the French actually at this point in the Louis Pasteur Institute, who said, it's not Prontosil that's effective, it's sulfonamide. And so they patented sulfonamide instead and made it widely available. And so that was technological innovation. Sometimes, of course, is driven by spite. That's just one example. Now the story doesn't end there either. Domcock was actually going to be awarded the Nobel Prize in, I believe, 1939, which may ring some bells. Mind if you heard. The Nazis were in power, and they actually denied, Hitler himself denied Domcock receiving the Nobel Prize. And so it was only in, I believe, 1947 that he was able to get the Nobel Prize officially. And he went on to have a more varied career. He worked on tuberculosis and died at like 68. He died at 68 of appendicitis because he asked a medical student to take out his appendix instead of like a regular doctor. And this operation wasn't successful. So that's one way to go. But he had a very storied career. He accomplished much, and he really produced the very first antibacterial drug. And really, at this point, it was a lifesaver. And to the point where it became a fad. Everyone started to take it when they had sniffles, and it eventually lost its efficacy because it became so widely used. But then we got penicillin, so no harm, no foul, I suppose. So this is one big way I feel that sulfur is implicated in our life, but there are other ways as well. And one of those other ways is in the field of chemosynthesis. These are bacteria that are able to use sulfur in order to generate their own energy. So you may see this equation a little bit reminiscent of something you may have seen earlier, of cellular respiration. But it's not quite the same, because you'll see here H2S instead of where we would normally find H2O. Or H2O would be a product and O2 would be going in. So we would see carbon dioxide as well on this side. So there are some differences between these two between cellular respiration and between chemosynthesis. But the biggest point is that sulfur is actually what takes the electrons at the end of the electron transfer chain for chemosynthesis. And so this is one way that we see elemental sulfur, pure sulfur, being produced in areas like hydrothermal vents. And we see at the very far corner there the example of a black smoker. It's a hydrothermal vent that we discussed a few weeks ago about where life may have originated. And interestingly, it's a whole ecosystem that forms around these black smokers. And these in the middle here, we have an image of tube worms that actually, instead of having a gut, they have a digestive organ that contains the bacteria that perform chemosynthesis. So it's a form of symbiosis where they've completely replaced like what we would think to be a completely necessary organ by forming a symbiotic relationship with the bacteria that are able to produce energy. So these were what we've discussed so far, just a few like interesting places where sulfur is used. But now I want to take a little bit of a deeper dive into amino acids, actually, amino acid containing sulfur compounds. And we'll actually take this pathway all the way to our trace elements. And we start with the two sulfur containing amino acids. Now, I say cysteine and methionine, but careful about cysteine, because this is not cysteine, this is cytosine, the nucleic acid. This is not cysteine, this is cytidine, the RNA nucleotide. This is not cysteine, this is cysteine, the dimer version of cysteine. And keep in mind the structure is pretty similar to cysteine that we'll see in a second. Nor is this cysteine, this is cysteine, the chapel. I don't know why there's so many different molecules that have very similar nomenclature here, but it is how it is, as well as just locations, I suppose. No, this is cysteine. And so these are cysteine and methionine respectively, and you'll see that they have a very similar structure. Cysteine, of course, has the, both amino acids, like all amino acids, contain the amino group, which is the carbon backbone, the carboxyl group right here, as well as a nitrogen-containing amino group, or amine group, that together combine to form the amino group. But and the way amino acids differ, as we've discussed previously, is what they have attached to the central carbon. And so in methionine, we have a few different carbons with a sulfur attached to two different carbons called a thiol. And here we just have sulfur at the end of the chain here. Now these are not, if you ever take a biochem course or you take a biology course, this is actually more likely to happen in a biology course, and you see on a test how many sulfur-containing amino acids are there. The answer that they probably want to put down is 2. That is not, the way the question is worded, that's actually not truly correct. Because there are many different amino acids that we don't normally think of in our 22 core amino acids. There are amino acids that aren't used for forming proteins, so the proteinogenic amino acids. And so unfortunately, our wording on tests can be a little problematic, maybe just not as precise as we like, but keep in mind that even though we're going to focus on these 2 amino acids, there are other sulfur-containing amino acids that are essential for us, and they go a long way towards the rest of our body, except for the sole reason that we are focusing on these 2, in addition to their implications in vitamins that we'll discuss in a second, is because they actually get formed into proteins. Now, also another important distinction between these two amino acids here is that methionine is what's called an essential amino acid. Now that doesn't mean methionine is somehow like more potent, like if you eat more methionine, like you'll become like superhuman or anything. And it's not the case. Both are found in pretty decent amounts in normal protein-containing compounds, meat, legumes, anywhere you'd find protein, you'll find these 2. But what is meant by that essential moniker is that we can make cysteine out of methionine, but we can't make methionine out of cysteine. So our body requires a few amino acids that it cannot synthesize. It requires it from nutritional sources, but there are other amino acids that it's able to synthesize, by itself, given that it has ample stores of those essential amino acids. So both are incredibly important, and we'll touch on a few different ways that they perform functions outside of just being part of proteins. But it's just something to keep in mind, that we can form cysteine out of methionine, but not the other way around. So why are these amino acids so important? Well, let's talk about cysteine first. So cysteine, because it has a sulfur at the edge of its structure of the R group, it's able to form what's called disulfide bonds. Now these are really, really strong covalent bonds that are able to, when we put them into a protein, are able to stabilize its structure. So we've discussed this a few weeks ago, but just as a refresher, proteins have different levels of structure. A primary structure is just the sequence of amino acids in order. The secondary structure is associations between the amino groups of those amino acids. They form different sheets as well as more helical structures. But then we go to tertiary structures, which is really a more robust folding and interactions between the side R groups of the amino acids. And that's where we see these disulfide bonds. Because a lot of the associations in the tertiary structures are really charged molecules interacting with each other. But you can imagine that these are powerful in the right circumstances, in the right environment, but that if that environment changes, they may be malleable, they may not form the right way. But if you have something locked down with a covalent bond, a disulfide bond, it really is solid. And even if you add a little bit of acid, yeah, it might change the other components of the molecule. It might cause a difference in the folding because of the interactions with other R groups. But these disulfide bonds will try their hardest to stay as they are, and so they really provide very stable structure to the amino acids. So this was cysteine, I should say. If I introduce so many different versions, I should be careful about my own nomenclature. But now let's talk about methionine. And I feel that methionine, this is a little bit more of a deep dive that we'll be taking, and we'll end at cobalt and vitamin B12. But methionine is one of the most interesting amino acids, in my opinion. And I feel like the discussion that we're going to have today is going to go into some depth. There is going to be some biochemistry. Just a warning here, there will be some diagrams that have too much information on them, but we're going to go through them step by step, and I'll be sure to point out what is relevant and what isn't. I always, of course, try my best to find the simplest version of the diagram. You can find sometimes in biochemistry that simplest is not quite simple, but we'll get there in a second. So let's start with something called methyltransferases, as well as a molecule called SAMe. Now SAMe goes by many different names. SAMe is actually the name that it's used in nutritional stores. You'll see it if you go to Costco. You'll see it as a nutritional supplement, but it goes by many names. It goes by SAM. It goes by as well as adomet, because that's probably closest to what I'm going to call it, because it really is adenylyl methionine. It's a compound with that starts with ATP, as we discussed earlier, the energy currency of the cell, but it also has this amino acid here, methionine. And we see already why we're discussing this, because sulfur, the chemistry of sulfur, is kind of the reason why we're able to have this structure form. We see here that sulfur has an incomplete octet. Now, it has a positive charge next to it because it only has 3 bonds, one to methionine and 2 to carbons as well. That bond to methionine is also a carbon bond, I believe. But if it had been a more electronegative element, that may not have been possible. If sulfur had been more electronegative, it would have demanded that it has a full octet. It would have been very unstable in this form. And now, keep in mind, of course, this isn't very stable as it is. It is stabilized by other things in the cells that allow it to stay in this form. But it's stable enough for it to engage in the reactions that it needs to. So that's another reason why sulfur is really important, because it's able to form these extra bonds. It's able to go to a super complete or expand its octet, but it's also stable enough where it's able to take an incomplete octet as well. So why am I bringing up SAMe? Because SAMe is one of the ways that a reaction called a methyl transfer occurs in our cells. So methyl transfers, which are catalyzed by methyltransferases, the nomenclature for the enzyme, is a very prevalent reaction in our bodies. They're really used in a variety of ways, but most often, and perhaps most notable, they're used in DNA synthesis. So a cell cannot replicate without methyltransferase close by. And this is actually the reason why the antibacterial agent we discussed earlier, sulfonamide, is able to work. Because sulfonamide is able to interrupt the DNA synthesis in bacteria. And so it's able to prevent them from continuing to grow. Because sulfonamide interrupts this pathway that we're going to be discussing over in the next few slides. So it's incredibly important, and when we see any deficiencies in the pathways and the molecules involved in the pathway that I'm going to discuss, we see anemia most often in individuals, but if the deficiency continues, we really see widespread effects throughout the body. This is an essential pathway in our body. Without it, we would not be the way we are. So I'm going to take a step here before we go deeper into the ADOMET pathway, into sulfur in general. Are there any questions with what we've discussed so far? All right. But remember, feel free to ask any questions even in the middle of the slide, etc. All right, let's continue. So before we can really discuss the importance of ADOMET and the importance of this pathway that involves sulfur, involves vitamins as well, we actually have to take a bit of a tangent and talk about coordination chemistry. Because we've talked so far about sulfur and how it's able to have many bonds, but this isn't something that's unique to sulfur. This is in fact very often in metal elements we see this type of interaction occurring. Now, a coordinate bond is essentially just a bond where one atom is sharing its electrons with the other atom. Now, this is separate from a normal covalent bond where we have two atoms sharing them, maybe not identically, maybe there's some polarization, maybe one is sharing more than the other, but there's, in a normal covalent bond, both atoms share. In a coordinated covalent bond, only one atom is able to share all the electrons required to make the bond. So we see this. I know that in previous lectures, I've shown you diagrams where there's a metal ion in the middle of a larger structure. Those are often because of these coordinate bonds that are holding that metal ion in place. And now that metal ion is now in a specific orientation where it's able to form other bonds, it's able to play its role regardless. And I think this diagram here goes to show, and I really like this diagram because it shows how we would normally think on paper in a chemistry class about our electrons, which just are orbiting around our oxygen, but also in three-dimensional space, how they would almost form a cage around the metal ion of choice, this time scandium, not Scandinavian, sorry. Now, why am I taking this tangent? Because we are able to now use this knowledge, use our sulfur knowledge, use our coordinate chemistry knowledge to go directly into vitamin B12. Now, vitamin B12 I've introduced to you earlier. I talked about it in my first lecture and a few lectures after that because this really set me on the path towards creating this class and this slide right here really. Because at the very center of vitamin B12 we have cobalt ion, and it's this cobalt ion that's held in place through coordination chemistry, through its associations with nitrogen and through the other nitrogen all the way down as well, that it's able to play its role. Now, we've discussed this earlier, but just as a refresher, vitamin B12, also known as cobalamin, or cobalamin, it has an A that I always miss in there, is an essential vitamin in our body. Even though it's number 12, there's actually not 12 B vitamins, because some of them were named and then found to be like just another version of the same one that was already named. There are some that aren't essential at all, but some of their names may be familiar, such as niacin, as well as folate, which we'll discuss both of those in a little bit. That is, I believe, vitamin B3 or vitamin B6, one or the other. Just take your B vitamins, especially vitamin B12. If you are vegetarian, like I am, you may find it difficult, and more so in vegans actually, you may find it difficult to get your normal daily allotment of vitamin B12, so it's recommended, especially if you're vegan, to take a supplement for that as well. It's very rare to be found in plant-based sources, but if you eat eggs, eggs have vitamin B12, milk has a little bit of vitamin B12 as well. Now, vitamin B12 is able to take many different forms, and this is how it interacts with sulfur. This is where both of these types of chemistry that we've been talking about combine in order to form products, and that's through forming associations here with another gaseous molecule. So now this molecule can be a few different things, and when it is, cobalamin takes on many different names. if it forms an association with a methyl group, as we were talking about earlier with adomet, that's that pathway right there. It's called methyl cobalamin. If it interacts with oxygen, it's called hydroxycobalamin. And actually, when it interacts with cyanide, because it can interact with cyanide, it's called cyanocobalamin. And this is actually the form in which you're most likely to see cobalamin if you are buying vitamin B12 off the shelf. Now, the cyanide isn't that dangerous in that small amounts, but it can, cyanide of course is toxic, and we'll discuss that next week. But interestingly, the ingestion of cobalamin, ingestion of vitamin B12, is actually the preferred way to fight cyanide poison, because it's able to go into your body and take up that cyanide, sequester it, so that way it isn't interacting with the rest of your cells. Well, that's enough. Now, we're almost ready to discuss, to show the connection between sulfur and vitamin B12. But we're not quite there yet. But we still need another piece of additional knowledge, and that's in cofactors and coenzymes. So we've been talking this time, for a few weeks now. I brought up vitamin B12, but what does it mean to be a vitamin, right? So a vitamin is one form of a enzyme. It's one part of an enzyme that allows that enzyme to function. And there's a little bit of a strict definition here between cofactors and coenzymes. But in the simplest forms, a cofactor is a non-protein chemical compound or a metallic ion, such as the ones that we've shown earlier, that is required for an enzyme's role as a catalyst. So any cofactor, this wide umbrella term of cofactor, is any metal or non-protein biological molecule that allows an enzyme to do its thing. Now, this is extremely broad, right? So that's why we have further subdivisions. Coenzymes are similar to, are essentially the vitamins plus other additional molecules that are non-protein. They aren't made of amino acids, but they allow the larger proteins to do their job. Now, these are two examples right here. You may recognize that one as we just saw a few slides ago, adomet. Adomet is a coenzyme, it's a cofactor, because without it, several enzymes, including vitamin B12, will not be able to function properly. We also see here the importance of sulfur again in these sulfur iron clusters. These are basically ubiquitous in our body. They're used in a variety of different enzymes, including the ones responsible for moving electrons along the electron transport chain. They're responsible in enzymes that are part of our cellular respiration cycle, and they're essentially found throughout our body because of those crucial roles. But without these two simple cofactors, those enzymes wouldn't be functional. Now, they're not always simple though. We can see, in this mess of a diagram here, we can see that many cofactors often have to work together in order for a desired product to be achieved. So what we're looking at here, we're actually in the edge of a mitochondria, and it's way too much detail, obviously, but what this diagram goes to show us is how important it is for cofactors and proteins to work together. One can't be functional without the other. So if you imagine here, this giant enzyme here is succinate dehydrogenase. You don't have to remember that, obviously, but it's essentially trying to create energy for the cell. It's part of the electron transport chain. It's also part of the citric acid cycle. But without the protein structure here, if we only had the cofactors, they would just be floating around. There would be no efficient transfer of electrons. You would hardly see any reactions. The efficiency would be close to zero, probably. On the flip side, if we had this giant protein that some poor scientists probably spent half their life trying to figure out the structure for, if we had this protein without the cofactors that we're seeing highlighted here, including the heme group, including other groups, if we had them, then the protein would be non-functional. It would have all the structure it needed, but it wouldn't have any of the actual crucial elements that transfer the electrons, that catalyze the reaction. And so both of these halves, both the protein and the cofactors, have to work together before we have a functional enzyme. So we've discussed now sulfur chemistry, we've discussed vitamin B12, introduced vitamin B12. Let's put it all together. Let's see what matters. Now, this next diagram is complex, once again, and we'll be going through it step by step, so keep that in mind. This is where the business happens. This is the cycle where we see sulfur chemistry interacting with vitamin B12. And without this cycle, we're basically the most common cited disease when we have a deficiency in this cycle, when something breaks down, and we'll discuss how it can break down, is something called pernicious anemia, where your blood cells are bloated because, as we mentioned earlier, vitamin B12 is really important for DNA synthesis. So if we can't have replication, our red blood cells eventually end up damaged, and that's the way, whenever this cycle breaks down, it most often shows itself in an individual as pernicious anemia. Now let's go through this step by step, and I've highlighted some of the things that we've seen before with green arrows, and we'll use those green arrows to kind of chart our cycle through. So we start with methionine, our essential amino acid, right? And methionine here is actually an end product. It's not an input, it's an output, because it's being produced by a smaller version of the molecule, one that's lacking this carbon group right here, called homocysteine. Remember when I said earlier that there's more amino acids than what we normally talk about when our 22 amino acids that get put into proteins? Homocysteine is one of those. Homocysteine exists for the sole purpose to engage in this cycle. And this cycle exists for the sole purpose of getting this methyl group moving. It's basically like a methyl group economy, where we need to have continuous transfer in order to get DNA synthesis going, because DNA needs to be going in many cells throughout our body all the time, right? If we ever have a blockage, if we ever get something that's not able to be regenerated in this whole cycle that keeps this methyl group moving, then things start to accumulate, things start to go wrong. And so the most often where this can go wrong is right here with vitamin B12. Say you have a diet that is deficient in vitamin B12. Say you're a vegan and you didn't take those nutritional supplements. Not saying that all vegans, of course, are deficient in vitamin B12. I, as a vegetarian, had low vitamin B12, so that was like my wake-up call that I need to start eating supplements. But say you go get your blood tested, regardless of what your diet looks like, and you're low on vitamin B12. Well, you're actually probably fine in the short term, because our liver contains, I believe, 3 to 5 years of a stored up vitamin B12 to keep us protected against the negative side effects of this. So it's only when you have vitamin deficiency over a long period of time that we'll actually see the effects that I'm going to discuss. And I think this goes to show how important this cycle is, that our body is taking up storage space that could be used for energy, that could be used for carbohydrates, glycogen, and it's using it instead to store vitamin B12. And that just shows how important it is to have this information. Well, vitamin B12 is going to be ferrying this methyl group that we've been talking about. And the way that it ferries it is actually by putting it back on methionine on one reaction, and actually taking it away from a different vitamin called folate. Folate or niacin, you may have seen if you looked at it like a box of Cheerios maybe, because often many cereals are actually fortified with folate because it's such an essential nutrient in basically general life. It's actually in one population where it's incredibly important is in expectant mothers, or even mothers who do not know that they're pregnant yet, because it's one of the most common congenital birth defects, even though I don't believe, unfortunately, that the fetus survives this defect many times, is a neural deficit, where the neural tube at the very first few weeks of embryonic development doesn't close fully, and so children that are born with this defect, often I believe spinal bifida is the pronunciation of the condition, where essentially their nervous system, their neural tube hasn't completely closed, and so their spine is open on one end. It's very fragile. Now, this deep birth defect is actually 50% of the occurrence of this birth defect. It can be traced back to a lack of niacin in the diet of the mother during the very first few weeks of pregnancy, because the spinal cord closing is one of the first steps that happen during embryogenesis, I believe around week 6 even. And so this is a point where the mother may not even know that they're pregnant at this point. And so it's highly recommended for everyone, of course, to be getting enough folate in the diet, but especially for women who are attempting to be pregnant, because this is something where once they know they're pregnant, it might already be too late to start taking those folate supplements. So something to keep in mind for everyone. For good health, folate is very important, and it's important because it participates in this cycle. That it's able to take up this methyl group right here It turned itself from that methyl group that it's able to get from other sources, it's able to convert it back into the regular folate. And if we have an accumulation there, this whole cycle that's turning can stop functioning. It's like cogs that have separated. And we can get an accumulation of cobalamin, and we see that we get an accumulation of homocysteine here. And we're not able to keep that methyl group moving. We're not able to continue with DNA synthesis. Now, where does ADOMET come into play? We introduced, ADOMET was the first one I introduced, right? Because of its intersection with the sulfur chemistry as well as our vitamin here. Well, ADOMET gives its methyl group into the cycle. It's one of the first ways that the methyl group comes in, because remember, ADOMET is just methionine plus adenosine, the same adenosine that's used in ATP. And so that over there is converted to adohomocysteine, just like homocysteine is here, and that cycle also continues. So there is the whole idea behind taking ADOMET as a nutritional supplement is that you can kind of get this this cycle supercharged. So even though there's not much evidence that that's very efficacious. Of course, the nutritional companies will want you to buy whatever they want you to buy, so that's that. It has been shown that ADOMET is actually useful as a painkiller. It has less side effects than the NSAIDs, we call them, Advil, etc. But it also has some side effects as well. It's most often taken with patients for osteoarthritis, but obviously, consult your doctor if you're in pain. Don't take advice from me. Don't take nutritional advice from me, please. Other than me telling you, please make sure your cobalamin and folate are good, because otherwise that can be a pain down the line. So that's essentially the key factors in this cycle. In this whole diagram, we've already talked about methionine, and now we see where methionine comes into play. And it's ADOMET going to homocysteine, and homocysteine going to methionine right here. It's one of those things, it's the input that keeps us going. And when we take this methionine, we can put it back in to ADOMET, or we can use it to make proteins as well. We see vitamin B12 here. Its sole function, it's actually, despite being so essential in our body, vitamin B12 is only used in 2 reactions, and you're looking at them. So its importance really goes to show that even 1 or 2 reactions, if they're disrupted in your body, can lead to disastrous results. And then, of course, folate as well. Folate is actually more varied. So it is used here in this cycle as well, but it also catalyzes other reactions. It's actually used throughout our body to catalyze a bunch of different metabolic reactions as well. Are there any questions about this diagram? Anything that you feel I brought up but I didn't quite pay off yet? Anything like that? So more generally then, this is the last slide for sulfur. Any questions about sulfur and sulfur chemistry? And so we have a few minutes left, and so I'm going to talk about 2 more elements, 2 more or 3 more maybe, depending on how much time we have, trace elements and how they're used in our bodies. Because I believe that we all have the tools now to understand what's going on in these diagrams right here. We talked about coordination chemistry. And that's really what these metal trace elements are doing. They're taking up a spot in an enzyme and acting as a cofactor to allow that enzyme to do what it needs to do. And so a lot of the trace elements that you all have chosen for your projects are also metals. And so I encourage you, when you start doing your research, to look more deeply about what enzymes it's being used as a cofactor for, and what those enzymes, if disrupted, might produce, what harmful effects they may produce in our body. Now, iron and copper are, iron definitely is most famous for its use in hemoglobin to allow us to transport oxygen to all the tissues of our body, but it's actually not that specific. It doesn't just carry oxygen. It can actually also is responsible for carrying about 20% of the carbon dioxide that's in our bloodstream as well. The rest of the carbon dioxide, of course, is dissolved into our blood, and so it's able to produce carbonic acid, or yeah, I believe that's the case, and it buffers that blood a little bit, but the rest of the carbon dioxide is actually attached to this heme because it's not that specific. And we talked, another non-specific case that we talked about last week, I believe, was rubisco, where we have a metal ion in its center, but it's able to transfer that ion out for other gases, not just the CO2 that it wants, but also carbon monoxide and carbon dioxide. And carbon monoxide actually can also be attached by the heme as well. And so it's used in cases, it can be used to treat carbon monoxide poisoning. Now that's just for vertebrates. That's the transfer molecule of choice for vertebrates. But these other molecules that are depicted here go to show that the same thing can evolve throughout the history of life. The same kind of mechanism for carrying oxygen or carrying gases throughout our organ systems can be produced in other ways. And one of those ways is by using copper instead of iron. So copper here in many mollusks, in fact all mollusks, so that's octopuses, I depicted here cuttlefish, but all like snails and things like that, everything under that category has what's called hemocyanin, where it's very similar to our hemoglobin, but instead it has copper as that chief element instead. Now this may remind you of something called, if you may have heard this word before, called hemolymph. Hemolymph is in insects like flies. It's actually not quite blood because it doesn't have cells that are responsible for carrying out oxygen. A lot of organisms are so small that they can get enough oxygen to their cells just through diffusion from the air. But that may be another place that you see the prefix of hemo. Now the other two you can take a look at them when this lecture is posted, but the other two molecules are just derivatives of these for other organisms, such as segmented, the green blood. I didn't know organisms had green blood until I put together this presentation, but some organisms of segmented worms have green blood, as well as some have purple blood or violet blood as well. Those are, I believe this is called a peanut worm. This is the violet blood is considered the most rare of these. So with that, we have 3 minutes left, so I'm going to end our lecture there. If you'd like, you can learn more about chromium and insulin in the lecture notes that are right attached to the PowerPoint. But let's spend a few minutes talking about the presentation, as well as looking forward to next week. Next week we'll be talking about poisons, and so I'll introduce this guy, Periclesius, also known as the father of toxicology. So with that, I'll end the presentation.